Okay, so this is a little bit of an extension. It's not strictly, it's not an A-level specification, but it's fairly interesting and something that you can understand. It's Multiplex Ligation Probe Amplification, MLPA. Now this is used very frequently to test for, if you want to test somebody who is a carrier for a genetic disease. So this is where the disease is recessive, and you only have the disease if you have 2 bad copies of the gene. If you're heterozygous, then you're going to be healthy. So for cystic fibrosis would be a very good example of this. Cystic fibrosis is a recessive disease. Sickle cell anemia, also sickle cell disease. So that's a mutation to the hemoglobin gene. And tay-sachs disease, which is a neurological degenerative disorder. They're all recessive, so you need 2 bad copies of the gene in order to have the disease. If you've got 1 copy, you're relatively healthy. Okay, so how do you test people for that? Now the problem is, if you go looking for the presence of the good gene, well, they will have it. And so, that's not really an approach. So if you think about the ways in which you could possibly test for these diseases, let's think about cystic fibrosis. So, if you remember GCSE, you probably draw little diagrams like that. If somebody was CC, they'd be healthy. If someone was that, they'd have 1 bad copy. They'd be a carrier, but they'd be healthier. And if they were 2 little copies, they would have cystic fibrosis. So the problem is we've got to develop a genetic test that can distinguish between these 2, right, which is difficult. So if you think about possible ways in which you could do it, you could maybe get the DNA, okay, so you get some tissue from a blood sample or something, extract the DNA. Digest the DNA with a restriction fragment, restriction enzyme, into little bits, and then you could do transfer to a nitrocellulose filter. So that's called a southern blot. So that goes onto a filter, and then you could test the filter with a probe. So test the DNA fragments with a probe, and so your probe, you would make, here's the probe, and you make this complementary to the healthy CF gene. Well, imagine you had your piece of nitrocellulose there, okay, and then you've got your bit of DNA. You've got DNA there. Well, you would have, say, that is the healthy gene, and that is you have 2 copies. So even though you've got the unhealthy gene there, your probe would bind, and you think, oh, I've got the healthy gene, wouldn't tell you anything. So you need to know, have you got 1 bad copy there? You can maybe think you could do it with PCR. Okay, so let's think how you could do that with PCR. You could, this is your cystic fibrosis gene, say, between those 2 green spots there, that's your cystic fibrosis gene. Now, if you've got a mutation of that gene, most mutations of cystic fibrosis, it's only a matter of a couple of nucleotides which were affected. There are somewhere that you get, and we'll put that there. So in some cases, it's as simple as 1 nucleotide which has been substituted, and that's enough, to turn the codon into a stop codon or something, and so you've got a short protein. So there's actually very little difference in the DNA sequence there. So if you put your primers there, your primer, the and then you would get, that would be your forward primer, and so you'd copy there, separate it out. So it's down there now. Put your reverse primer there. Make a copy. Well, and then you'd have to run that on a gel, and to see you'd end up getting this expected this length of fragment. Well, it would be exactly the same for the disease gene here, and for the for a gene with no mutation in it. There may be 1 or 2 base pairs difference maximum. That is not going to tell you anything. So obviously a different approach is needed. Okay, so let's get rid of this. What happens is then we're going to use this this new method, this MLPA. The clue is in the name, really. Not really what multiplex mean, but ligation means. Remember what DNA ligase does? It joins 2 fragments together. It will join 2 bits of DNA together. Okay, so you're going to do something which involves joining 2 bits of DNA together, and then you're going to amplify it, those 2 joined bits, and amplify it using PCR, and then you're going to look for that amplified fragment using electrophoresis. So let's see how that works. Okay, so here is our gene, and we'll say this is our normal gene here, and on this side I'm going to draw, this is the mutated version of the gene. I will draw the extreme example, the hardest possible thing to tell the difference with there, and that's where you have a single base substitution there. Right, so what I use, I use 2 different primers. Okay, so you get 1 primer, I'll draw it in red there, which ligates, which will hybridize to that section of the gene. So it's complementary sequence there, and you get the blue one, which is right next door to it. So there's no gap at all there. That continuous sequence, but you have got 2 separate bits of DNA. And then usually what's on the end of these is called, charmingly, it's called stuffer DNA, stuffer, and that's just to make it a little bit longer. Those green bits, they don't complementary base pair to the gene. So this is the CF gene up here. Sorry, this black line is the CF gene. So you need to make the before you do anything, you need to make the DNA from the patient, single-stranded. You have to denature it, get it single-stranded, and then you hybridize your red primer and your blue primer there. Now, in the person who has got the mutation, right, the red will go there, but the blue won't quite join on properly because one of the bases, it means it's just not quite, it's lifted off the surface. And then what is added then is this enzyme DNA ligase. DNA ligase will join 2 bits of DNA together. Now DNA ligase will work here. All right, so you're going to get 1 continuous piece of DNA there, but it won't work. The DNA ligase will not work here because the match has to be actually perfect between the 5' end of one and the 3' end of the other in order for them to join together. So they're not going to join together. So, and I should show you that's got the stuffer or the filler DNA there as well, because they won't join together. So the next step in this procedure is to separate the strands, and that's done in the usual way by heating, and it's done in a PCR thermal cycler machine. Okay, so you will get your original bit of DNA, and you're going to get this by the way, I should say, usually this kind of sequence here, from there to there, is about 80 base pairs long. So it's only a small portion of the gene. The gene is going to be like, you know, maybe 3,000 or 4,000 base pairs long. So we've got our ligated probe. So this is the ligation probe, but we have managed to ligate our probe. Now this is where it's a bit different from normal PCR, because normal PCR we would try and amplify the DNA, the gene, wouldn't we? But that's not what's going to happen here. We're going to amplify, not that one, that thing. We're going to amplify this ligated primer. Okay, so you need another, this is a normal PCR primer, so this, I'm going to draw that here. And that's going to be a fluorescent primer, that little bit there. Fluorescently labeled primer for PCR. And that's going to be the forward one. And we also going to need a reverse primer, the reverse primer. And that they don't bother fluorescently labeling that. You only need to label one of them. So we'll see what happens there if we do that then. All right, so we're going to ligate the primer, say there. This is the 5' end. And we had TAC polymerase, the heat sensitive polymerase, and that's going to fill in up to there. Then we're going to separate those two strands by thermal cycling. So I'll just move that down a bit. Let the primers anneal again. So the reverse primer is going to go on that one there, and the forward primer is going to go there again. Let 5' to 3' polymerization occur. That goes there. That goes there. Then we heat it and separate again. So I'll just move this one down a little bit here. Separate the strands. So I'll just rub that one out because that one is being moved downwards. I'm going to move this one downwards here. Okay, so we've got the black and the purple. Okay, so let's see now what happens when we do another cycle. So you're going to get the reverse primer going there, black, reverse primer, synthesize the new strand of DNA, reverse primer there, synthesize the new strand of DNA. Here we've got forward primer, and we're going to forward primer and make that there, and so on. Okay, so we're going to go from we've gone from one of them, we've got 2 there, and the next time it's going to be 4 and 8 and so on. Let's see what happens with the non-ligated primer. So this is the person who's got a mutate. This is the gene that is mutated, and don't forget, a person is going to have 2 genes. So you could have 2 healthy genes or 1 healthy, 1 mutated. Let's say this is the mutated gene. Well, when we separate this, when we do when we heat to separate the strands, all that's going to happen there is, right, that is going to go there, and the other bit is going to be completely separate from it. Now we add the first primer in there. The first primer, right, that will go there, and yet that can make a copy of that. Let's heat that up and separate them. And you can see this is now different, because in the next round, the reverse primer, it can't bind to that. That's the wrong sequence, so nothing happens with that. That's the end of the road for that bit of DNA. Yes, it can make another copy there, and that can happen every time. But on this side, the numbers are increasing 1, 2, 1, 2, 4, 8 geometrically, On this side, the numbers are just increasing 1, 2, 3, 4. Okay, so if you do 25 cycles of the non-ligated one, you're going to end up with 25 copies, which is hardly anything. With the ligated one, you're going to end up with 2 to the power of 25 copies, which is a lot. So you're going to get vast amounts of this. You're going to amplify the ligated primer, but the non-ligated will not be amplified. Okay, so all you've got to now, if you have got 2 copies, so if you've got 2 copies of the good gene, you're going to get 2 times as much of this amplified fragment made. If you've only got 1 copy, in other words you are a carrier, you will only get 1 times of the amount of the amplified, or half of the person who's got two copies. Now you need to have a little internal standard with this. So what is done is, if this is, if I just think this is the whole DNA, and let's say that this is our cystic fibrosis gene here, and this is where we're going to put our primers, which just run by the mutation, you know, the ones that won't get ligated. So put that, they go there, and don't forget you have this stuffer DNA stuff stuck to the side of it. Yeah. So that's our CF gene, and what is also chosen is another gene, one that isn't mutated. So let's say we've got another gene here, or it can be, it's probably best for it to be a gene rather than non-coding region, because genes tend to be more highly conserved, they're always the same. So you have another one here, another two primers which can be ligated, and they're going to be ligated in everybody, these ones, because this gene is not mutated. So this is the standard. Okay, so that will get ligated, so you'll end up with a ligated primer, and this is where the stuffer DNA comes in. You're going to make that's going to be a little bit long. Say that's like 800 kilobase pairs long. So this one is 800, and they use the stuffer gene slightly smaller for that one. So that one isn't going to be quite 800. Let's say that's going to be 600 long. So that will make a smaller fragment. This is only 600 base pairs long. Now, when you amplify this, you're going to, let's say we use a yellow fluorescent probe. So that's going to make yellow DNA. When you amplify this one, let's say we're going to use red, okay? You're going to get red fluorescent material. Okay, so let's see what happens when we run that in the gel. So it's a capillary electrophoresis. So here's a little thin capillary tube. The sample, after all the cycles of replication and so forth, the sample comes in this end. DNA always moves towards the positive voltage, so that's positive. And at the end here, we will have a laser shining UV light, and here we've got a detector. And that can detect either yellow color or red color. And the trace that we're going to get will be something like this. Now, we said, what did we say are, so let's say this is increasing size. We said that our, this ligated primer from the standard, we said that's a bit bigger, that's 800. Okay, so that's going to, and the other one's 600. So, right, so say we get a graph which looks like this. Okay, so the standard here and the CF are both the same peak height. Okay, so that means we've got the same amount of DNA in both of them. Now the standard, we will have, obviously we'll have 2 copies. So that means that's the healthy CF gene there. You've got 2 copies of the healthy gene. So that person isn't a carrier. Imagine if they were a carrier, then the results would look something like this. They'd only have half the amount of DNA from the amplification there, because only one of the genes would be good at being amplified, so you'd have the peak would be only half as big. So that person, you would realize, is only got 1 copy, so they must be heterozygous, they must be a carrier. And of course, if you put somebody's DNA in here who was suffering from the disease, they'd know anyway, but you would find that there would be none of the yellow amplified fragment made at all, because you wouldn't get ligation with either of the primers. These primers here, they wouldn't ligate there because there'd be a mismatch. The 2 ends wouldn't be close enough together, so nothing would happen. So just to remind you that that's why is it called ligation, because you get these 2 primers, the red one and the blue one, and they are right next to each other. They should be right next to each other if there's no mutation. If there's a mutation, there may be a little bit of overlap if there's one deleted, or if one of the bases isn't pairing right, or if there was an insertion, then they'd be too far apart. So you wouldn't get any ligation. The 2 ends would not stick together, and it would not be amplified. So that's the ligation probe amplification bit. The multiplex bit here is usually, when they carry out this, I've just done it with 2. I did the CF gene, or whatever you're interested in, and the standard. But very often they might do it with a whole load of different primers for different possible mutations. The CF gene in particular there's a lot of different mutations, so they could, I think there are about 30 common ones. So you could use 30 different sets of ligation primers, and you could find out exactly where, not only the homozygous and heterozygous, but you could tell which mutation they were affected by. Okay, so that's the way in which you can show up if a person's only got 1 copy of a gene, which is difficult to do because you've also got the good copy there, which is also going to, you're going to think you've got something there, because when you test for it, you do have 1 good copy. This is to show you've got 1 good copy and 1 bad copy.